Hello everyone, my name is Anukriti Singh and I will be briefly presenting my work on how miR-155 attenuates oncogenesis by targeting AGTR1, NF-kappa-B and CXCR4 axis in glioblastoma and breast cancer. This work was done at the Molecular Oncology Lab under the supervision of Dr. Bushra Ateeq at the Department of Biosciences and Bioengineering at IIT Kanpur and is published in the journal Neoplasia with Dr. Nidhi Srivastava and Ms. Anjali Yadav as co-authors. Cancer represents uncontrolled cell division which originates with an unwanted mutation and accumulation of defects in many classes of genes. Glioblastoma is the most aggressive brain cancer. Patients suffering from glioblastoma usually succumb to death in the first 15 months of diagnosis. And the basis of glioblastoma occurrence is still unclear. And the treatment regimen includes surgery followed by radio and chemotherapies. However, unfortunately, the tumor comes back after 6 to 9 months. Therefore, the development of new targeted treatments and therapeutic modalities is highly warranted. Angiotensin II receptor type 1 or AGTR1 is a gene responsible for regulating the blood pressure in human body. But when AGTR1 generates too much or overexpression of its protein, it drives cancer in a variety of organs such as cervix, ovary, breast, and prostate. Our study unravels the mechanism underlying the overexpression of AGTR1 in glioblastoma and breast cancer and suggests possible therapeutic approaches to target it in AGTR1 positive patients or patients with overexpression of AGTR1 in both breast cancer and glioblastoma. We discovered that excess levels of AGTR1 are present in glioblastoma and breast cancer patients. To confirm if AGTR1 was indeed the culprit, we silenced or decreased its levels in glioblastoma and breast cancer cell lines, and as expected, a reduction in oncogenic or cancer-causing properties of these cell lines was observed. We also found that a microRNA named miR-155 is involved in controlling the levels of AGTR1 in these cancers. Interestingly, we next introduced miR-155 in the AGTR1 positive glioblastoma and breast cancer cell lines. To our surprise, when the miR-155 levels in these cancers was increased, AGTR1 levels decreased, along with decrease in several oncogenic properties such as cell proliferation, invasion, and ability to form tumors in mice. We also performed an important experiment in chicken embryos, where formation of new blood vessels failed when glioblastoma cells with high miR-155 levels were implanted in the chicken embryos. More importantly, in my xenograft model, when we implanted AGTR1 positive glioblastoma cells modified by introducing miR-155 and unmodified glioblastoma cells or control cells in two groups of mice, we observed that tumors grew aggressively in the mice of the control group, whereas there was almost no tumor growth in the mice implanted with the cells having high miR-155 levels. Furthermore, we unraveled the interaction of AGTR1, NF-kappa-B and CXCR4, that is, the AGTR1, NF-kappa-B, CXCR4 signaling axis as a key player in mediating the overexpression of AGTR1. We have shown that a signaling pathway called NF-kappa-B is activated when AGTR1 gets stimulated, which leads to a chain reaction of activating another cancer-causing gene, CXCR4, and further upregulates AGTR1 by a feedback loop. Notably, we disrupted the signaling axis by using miR-155 or IKK inhibitor to abrogate oncogenesis in glioblastoma and breast cancer. Conclusively, our study proposes that the use of miR-155 replacement therapy or pharmacological inhibitors of IKK complex might be beneficial for the treatment of AGTR1 positive breast cancer and glioblastoma patients. Thank you for listening.